right, so the wrap-up for this morning is about methodology, which sounds kind of dry, but it's actually not at all. This talk was scheduled for an hour tomorrow, but actually Ed Louie, the head of the JAX lab, had time to talk, so this got shifted and compressed. So I'll do what I can. It previously was intended to involve all the faculty, but I'll see what I can do about it. The challenge for us all in this new field, the new parts of evolutionary applications in medicine, is to try to figure out how we can, demonstrate that we're really strongly figuring out which hypotheses are true and which ones are false, and how we can help people who don't know too much about the field to be considered and thoughtful and sensible about it. There's a huge tendency for people to try to make some generalization, like, in fact, I go to medical meetings and talk with heads of leading foundations, and they say, evolutionary medicine, oh, see, Gould and Lewontin said that we shouldn't do just those stories, so that's useless. And of course, they don't know anything about the field, and grand generalizations about all studies about adaptation, but that's what's out there a lot. And I think it's incumbent on all of us to figure out how we can be both cautious and careful in doing our research and also encourage creative kinds of thinking. There's a real trade-off here, and it's up to all of us in this room to help others to deal with that trade-off in a sensible way. I'm going to whiz through just some, I call it why is Darwinian medicine difficult, because I'm focusing on some of the ideas that George Williams and I have proposed. And especially this idea of asking a new question. It's not a new question, really. It's an old question about why bodies are vulnerable to disease. It's William Paley's question, but we're taking it seriously in a way it hasn't been taken before. Ernst Mayr makes very clear in his wonderful book, The Growth of Biological Thought, again, that there are these two different kinds of questions we ask. One are proximate questions about how things work, and also evolutionary causes. And you've noticed that the study of evolutionary causes is as legitimate a part of biology as is the study of the usually physical chemical proximate causes. So this is absolutely fundamental to everything, as you know. Here are the characters who made this all possible: Tinbergen, certainly, Ernst Mayr, who I just mentioned, John Maynard Smith, and my friend and recently deceased colleague, George Williams, without whom none of us would be here. Tinbergen, you all know his four questions. How many people know Tinbergen's four questions? Okay, so this is a problem. One of the great advances in biological understanding is Tinbergen writing an article to celebrate the aged birthday of his mentor, Konrad Lorenz. Konrad Lorenz had turned 60. Doesn't seem very old to me. Konrad Lorenz turned 60, and his student, Tinbergen, wrote this article on the aims and methods of ethology, where he talked about there being four different questions that biology needs to answer about any trait. One question is the ontogeny, the development within the individual. Another is what is the mechanism and how does it work. Another is the phylogeny of the trait. And the fourth is what selective advantage or other fitness consequences of the trait account for it being the way it is now. Tinbergen's article has by now dramatically eclipsed any work that Lorenz ever did, and is the foundation for almost all work in animal behavior, and increasingly, gratefully, in the rest of biology as well, where people are recognizing that a complete understanding of every molecule and every connection and every lever and pulley doesn't tell you why it is that way. This is an entirely different category of questions. They're not competing. They're both there. When George Williams and I began our work, and I always mention this because it's hard for us to get our minds back in the perspective of people just starting out in this field, we spent 6 months asking each other, so what is the evolutionary advantage of cancer? And what is the evolutionary explanation for heart disease? And we kept on trying to explain diseases in evolutionary terms, and finally we recognized that that's not a very good question. For a very simple reason, diseases are not shaped by natural selection. It's inappropriate to seek an evolutionary explanation for something that is not shaped by natural selection. The right question is, why do bodies have traits that seem suboptimal in ways that increase vulnerability to disease? So at least with this part of evolutionary medicine, we're not trying to understand diseases, we're trying to understand traits that leave us vulnerable to diseases. Again, what I was taught in medical school is natural selection is just too weak. You can't expect some random process to make bodies any better than they are, and that's absolutely true. There are many aspects of our bodies that are limited by that, but as George and I worked together, we recognized that there are some very systematic kinds of explanations evolution offered, not only for why bodies work so well, but also for why bodies have traits that leave them vulnerable to disease. So this is, I would emphasize very strongly, only one small part of evolutionary contributions to medicine. There's all these other things, most of which are the focus of what you're hearing here, like phylogenies and the like for bacteria and for tumor cells. A very different kind of approach, watching things small evolve more quickly. But these kind of questions have engaged a lot of enthusiasm and energy and interest amongst people, with huge benefits and pretty big costs also. Those 6 reasons, you probably all know this: must match with environments, competition with pathogens, every trait is a trade-off, constraints on natural selection, organisms are shaped for reproductive success, not health, and defenses. We're not going to go into any of those now. And that's not a magical list, but it has turned out to be very useful for people to organize their thinking about the kinds of possible explanations for why organisms aren't better designed. There are huge advances, and some of the books that got things going, Steve's book from the Sion meeting, and what's wonderful is that this is in a 2nd edition from Oxford. This is also a 2nd edition from Oxford, and the definitive text for right now about the field. This is a textbook that Peter Gluckman has come out with, a 2nd edition of which is due fairly soon. This is a book auf Deutsch. So things are coming along pretty well. The special issue of PNAS was also a milestone, although it's only one. We're really still just getting started. But here we come to the pause point, where there's a lot of interesting things going on, but even sophisticated scientists and journalists often have no idea about how to really go about judging the correctness of these hypotheses. And my supposition is that there's several reasons for this. Here's one good example I use often as kind of a whipping boy. This is an article that appeared in a medical journal about explaining diabetes type 1. And the editor of a major newspaper called me up and said, the evolutionary explanation for diabetes has been discovered. I said, wow, cool, tell me. They said, well, turns out that high glucose levels in your tissue protect against tissue damage from freezing, and the genes for diabetes turn out to be about the same age as the Ice Age, and they're more common in Northern Europe, and therefore diabetes type 1 is a protection against, was shaped to protect against the Ice Age. And of course I said, that's ridiculous. Don't waste your time on that. That's diabetes type 1, by the way, kills you, which is really bad for your fitness. And it's only a few people in the population, not everybody. I mean, just don't spend your time on this. But I got a nice note from that person a few weeks later saying, Dr. Nesse, we respect your opinion, but we've consulted 3 of the leading diabetes experts in the world, and they all assure us that this is probably the correct explanation. So this is a leading journalist from a well-respected paper, and this person, I like and admire, but she had no idea about how to go about actually doing an assessment. And I said to her, I didn't tell you to check with 3 diabetes experts. I said, check with 3 evolutionary biologists. And the diabetes experts just don't know much about methodology in our field. So the theme, this is really hard science. Again, to emphasize that what I'm talking about now is a small subpart of evolutionary applications. These areas are much better developed: infectious studies, phylogenies, and evolutionary genetics. Lots to be done, as you've been hearing, but much better developed. So again, why is Darwinian medicine difficult? Much is just ordinary difficulty, using the methods we've just been talking about, good hard science. But I think human cognition leaves us especially vulnerable to making mistakes when we try to do thinking carefully about functions and the evolution of vulnerability traits. First of all, our minds are fascinated by paradoxes. We love it when somebody says, the appendix is actually good for you. I mean, it's a mystery, it engages a mystery part of our brains. And we love simple monocausal explanations, of which it's very easy to come up with lots of them in our field. And we love functional explanations because it gives a sense of purpose and meaning to things that's missing from a lot of mechanistic explanations. And then there's this unfortunate tendency in humans to grab onto one explanation and make it yours and defend it like a flag or something like that, and not be able to allow yourself to seriously consider other kinds of hypotheses. You'll notice I'm not going to do the long argument here, but one could make an argument that these are evolutionary explanations for why we have difficulty doing evolutionary explanations. So what are the difficulties we face? In having many student papers that I've tried very hard to help them be careful about, and even at the end of the semester I get papers saying the evolutionary advantage of schizophrenia is, and I just shake my head in disbelief and say, I failed. I tried my very best with these students and I failed. What could I have done differently to help them think more clearly about this? They often don't have the right objective explanation. They're not specific about the kind of explanation or the possible answers. Oftentimes people get the misimpression that one kind of evolutionary explanation is correct in contrast to others when many may actually apply. And exactly what methods to use are another problem. So what are the solutions? The solutions are to try to get people to be perfectly exact. Could I ask you to pass these out, please? To be perfectly exact about what you're trying to explain about, it's just standard science, right? Just be perfectly clear about everything. But it turns out to be really hard in this area. Here's why it's hard. There are at least 10 different areas where we're doing work. And Steve Stearns and I worked together at length to write a paper about opportunities in evolution and medicine and emphasize that there are these two different kinds of questions even within evolution, one phylogenetic questions and the other questions about function, and 5 different objects of explanation. An excessively busy slide, but I keep it over my desk to help me think clearly about an article that I'm reading. And when I'm reviewing articles, I try to ask myself, are these people asking about phylogeny or function? And is the object of explanation a gene or a trait or a pathogen trait? Because you use different kinds of approaches depending on which of these 10 boxes you're in. I already mentioned the 6 different kinds of vulnerabilities, so we won't take time on that. And then we have all these different methods that we can use. People say, well, how do you test evolutionary hypotheses, as if you want one method? But in fact, we have a lot of different methods that we have accessible to us, and we don't use just one. We use everything we can. Consistency with theory, modeling for plausibility. Comparative method is the strongest when you can use it, and usually for humans you can't. Comparative methods are subpopulations, epidemiology, mapping form to function, experimental manipulation, natural experiments, and watching evolution happen. The first and biggest error that I find everywhere is people trying to explain diseases. Same error that George Williams and I made. Diseases do not have evolutionary explanations, mostly. Traits that leave us vulnerable to disease do. And again, there's this big tendency to explain rare traits and genes as if they are adaptations. So people will say the evolutionary explanation for schizophrenia is something, or the evolutionary explanation for hemophilia is something. But you know, that's not usually how we do it in evolution. We talk about a trait that's universal in the species and try to explain that. If we're trying to explain something that's rare in a species, it involves all kinds of complications. Again, the big mistake is trying to explain a disease directly. You'll see why this may be useful in some of your groups, because as your work groups get going, some of these issues are going to be coming up, and this might help you to dispense with them more quickly. So in my frustration after several years, I finally, thanks to Peter Gluckman prodding me repeatedly, wrote an article with 10 questions that you can ask yourself as you're working on a project about evolution medicine to help you avoid the simple mistakes. And I should say this started off as 10 mistakes a lot of people make. So this is the kind of positive version. The negative version is in a chapter I wrote in the Turathan volume. And I'm not going to go through, well, maybe I will go through these. You've got the copy in front of you so you can look at them. The big problems here, though, are what is it you're trying to explain exactly? And mostly what we're looking for is a trait that seems suboptimal. What kind of explanation do you want? Be really clear about whether you want an evolutionary explanation or approximate one, and whether it's a phylogenetic or a functional one. As for the alternatives, the big question we're always asking is what we're trying to figure out, is the trait an epiphenomenon or something else that's not been directly shaped by selection itself, or is it something that has indeed had its own variations influence fitness? And then what methods do you use? Examples. Some are pretty easy. Why do we have a blind spot in the eye? It's because of path dependence, and the fitness value you would have to go through to get to an eye that's like an octopus eye without a blind spot is impossible to get through. There would be no survival or success for individuals who are blind for 10,000 generations and no selection force to push them on that direction, so we're stuck with this. If we could find similar kinds of things in bacteria, it would be a wonderful kind of insight that would allow us to attack things that they couldn't escape very well. As for myopia, it seems easy. It's a heritable disease, and so we're looking for, and framing the question correctly is, why do genes for myopia persist? But as you look at it more carefully, some of you were in our discussion group yesterday, it becomes quite clear that we have a number of methods, epidemiology, theory, mismatch, and constraint. So it turns out even though this is highly heritable, that does not mean there are defective genes responsible for it. It means that these genes are interacting with environments in ways that cause disease, and they probably were not causing any myopia to speak of in ancestral environments. It's only when these genes interact with modern environments. And I have thought for some years, and the theme of what I'm going to do in the next 15 minutes is tell you a lot of things that I thought were right, and as I get into it more, I realize there are other alternative hypotheses that we need to consider, which is a challenge for all of us. So I was pretty sure it was early reading, good data on people in the Inuit places in Alaska, where there was very little nearsightedness in 1900, and a few years later, after reading came in, most of the kids, or 30% of the kids, same as elsewhere, were myopic. I thought, no, that does it for me. It's genetic vulnerability interacting with early reading explains myopia. But it turns out last year at this conference, Peter Ellison pointed out in an article where someone made a very strong argument that actually it's the availability of refined sugars that increases the growth of the eyeball, proportionate and nice epidemiological data mapping that. And I haven't gone to the trouble of really trying to think through which is right and how you determine them, but it's a case in point that I jumped to my first conclusion, I was happy with it. But I hadn't thought through the alternatives. Unfortunately, someone else has. Gout is a very nice one, how to frame the question. What's the evolutionary explanation for gout? That's not the right question. The right question is, why has natural selection left us humans with uric acid levels that are so high, resulting in some people getting those crystallized uric acid crystals in their big toes? And the theme here is that genes are trade-offs, and the method we use is the comparative method, looking across different primates at rates for, I should say the hypothesis first, is that uric acid is quite a fine antioxidant, and because it's an antioxidant, for a longer-lived species, natural selection might well have raised uric acid levels proportionate to where you get the maximum benefit with the minimal costs, despite the costs of precipitation of uric acid. And this is pretty good data from one of my students, looking at the lifespan on the y-axis and the uric acid levels on the other. Nice comparative study. But then you have to look at the proximate mechanism, and it turns out there's a specific defect in uricase in humans that's not present elsewhere. And this one gene change accounts for our high levels. Is that because that's been selected for, because it gives advantages, or is it some other accident of a bottleneck or something that's resulted? Another unanswered question I'd love to see somebody answer clearly. Why isn't the radius thicker? So that when you fall forward, it always breaks right there when you fall forward. So the framing of the question is, why isn't the radius thicker? Notice we're always framing these questions in terms of the trait we observe versus some trait we imagine that we think would be better. And in this case, I've been teaching for years that the trade-off with mobility is responsible. Very hard to actually find evidence that would confirm that. I suppose what you could do is measure people whose radii were thicker and thinner and look within populations at relative mobility and how hard you could throw a ball and the trade-offs involved. This is a very good story, I think, but it's very hard to confirm with data. One of my favorite examples is bilirubin. I've always wondered why there is bilirubin, given that it's a breakdown product of heme metabolism, of course, and biliverdin, its precursor, is more water soluble and less toxic. So why on earth does the body use energy to make a substance that's toxic and hard to excrete later? And again, in my work with George Williams, he said, well, actually, bilirubin is also a very nice antioxidant and might be especially useful for a longer-lived species. So how do you test this kind of thing? Are experimental methods ever appropriate in evolutionary medicine? And indeed, they are appropriate. In this case, Sedlak and Snyder were able to knock out a gene that allowed this cycle to go around between bilirubin and biliverdin, and those cells died very quickly from oxidative damage. So that's pretty nice confirmation experimentally of this hypothesis. Clinically, you can get further confirmation by looking at people who have variation for whatever reason in their serum bilirubin levels. And the people who have higher than normal bilirubin levels in middle-aged men have heart attack rates that are less than half of that of other men, confirming both the role of bilirubin and the role of oxidative damage in the coronary arteries. Alzheimer's disease is another huge mystery, and talk about something that's on the table right now. Lilly concluded a study last year that they've been doing for 3 years of a substance that interfered with the beta amyloid precursor protein being changed into beta amyloid. And everybody, and me too, is very hopeful, because beta amyloid is the substance that surrounds your nerves when you have Alzheimer's disease, very strong correlation. Everybody's very hopeful, but halfway through the study, they broke the code and found out that the people who'd been getting the drug were declining substantially faster than those who were not getting the drug. There are now several other trials going on with equal hopes, slightly different ways of interfering with beta amyloid synthesis, but it's, when you take an evolutionary view, nobody's really even thinking about why beta amyloid is there in the first place and how it's regulated. And this is one article from Socia that shows that beta amyloid is quite a nice antimicrobial. I don't know the significance of beta amyloid being an antimicrobial, but the evidence for it is quite good. Maybe there's infection or inflammation, or maybe who knows what connection is, but at least there's thinking going on, so far as I can tell, unconnected with anybody who's in the drug development area. Here's another one of my favorite hypotheses. A lot of people in East Asia can't drink alcohol because they get sick. Their aldehyde dehydrogenase doesn't work. Aldehydes build up in their system, and they get sick when they try to drink. And it's been very easy to say that in those areas, distillation of alcohol has been present much longer than other areas, and people who have this genetic abnormality would be protected from becoming alcoholics. A really good story, and I've always wondered, so what's the evidence? How are we going to test this one? Finally, about 6 or 7 years ago, and 10 years ago now, somebody did a study, genetic study, looking for where in the world you actually found these abnormalities, and this is what you find for East Asia. And specifically, they looked for haplotypes and found that this locus is in the center of the largest haplotype in East Asia, meaning that all of this chunk of DNA has swept through the population together. It's a very strong signal of selection, exactly surrounding this locus. And I thought to myself, okay, we've nailed this one. Finally, we really know that natural selection has increased the prevalence of this genetic so-called defect to protect people in that area from becoming alcoholics. And then I read another article by an anthropologist who pointed out that the geographical distribution of the aldehyde dehydrogenase deficiency in East Asia was not necessarily at all congruent with alcohol use patterns. It was more congruent with other dietary patterns. Might have something to do with cabbage or something like that. Again, each one of these turns out to be difficult. Steve?
Ken Kidd, who has done a lot of that work, has shown that, in fact, this haplotype is protective against the fungus that infects rice as a stored product.
Wonderful example. Thank you. I'd like that paper, please. But isn't it both fabulous and intimidating for each one of these things to see that you think you got it nailed, and there is evidence there's selection acting, but you really have to consider every single possible hypothesis. Cystic fibrosis is another classic one. A number of articles suggest that cystic fibrosis alleles are prevalent in Northern Europe because they, in fact, protect against death from dehydration from diarrhea. Two possible mechanisms. One is that they decrease the amount of fluid lost through the large bowel through cholera in mouse studies. And there's another study showing that Salmonella typhi can't penetrate the mucosa as well in heterozygous mice for the cystic fibrosis alleles. You know, those are both interesting. But does this really explain the relatively high prevalence of cystic fibrosis alleles? I'm not at all sure. I mean, all of these very rare genetic mutations need to be considered in terms of their fitness consequences. And our tendency is to say either they're adaptations or they're mutate or they're random and drift. But of course, you don't have to really choose between those. There are combinations of little tiny selection forces that may be acting in some subpopulations at times and drift that might account for something like this. I find a lot of people just automatically assume that cystic fibrosis, they say it's an adaptation. No, it's not. If it was really adaptive, it would be spreading worldwide quickly and it'd be universal. And then there's atherosclerosis, one of the most common devastating things that we all are trying to cope with. What's the evolutionary explanation? But we need to consider all possible hypotheses. A mismatch, we're eating diets that we were never intended to eat and our bodies can't cope, and inflammatory substances are much higher levels, trade-offs with inflammation, constraints and defenses. And I have Bill Aird to thank for, he's the fellow I mentioned who spends his summers up here and who made it possible for us to have this course here. Bill Aird has encouraged me to think about this and read a chapter for his endothelium book, and with another colleague who actually knows cardiology, we recognize that having the capacity for inflammation in the endothelium is very crucial for preventing pathogens from passing through at the cost of the risk of this kind of thing when you interact with modern environments. Case study here, multiple explanations can all be true at the same time. Dan Kruger and I have gotten a lot of publicity for our work on why males die younger than females. It's really all organisms where males compete with each other for mates. And we thought we had a fine, sufficient evolutionary explanation. But yesterday, somebody came out with an article on Drosophila showing that, in fact, mitochondrial mutations in females are selected against. Mitochondrial mutations that decrease female fitness in fruit flies are selected against because the mitochondria pass through females, but in males they're a dead end, and so there's not much selection against mitochondrial mutations that harm males. And they have evidence that in fruit flies, a substantial proportion of the increased death rate in male fruit flies is due to mitochondrial mutations. So you think you've got these things nailed down, and there's always one more thing to think about. Challenge for all of us is to actually allow them all in and think about them. Conclusions. It's wonderfully difficult, and I use the phrase wonderfully difficult because we shouldn't, just because this is hard, say we shouldn't be doing it. And some people say, oh, it's too hard. No, this is good science. That's why science is fun. It's wonderfully difficult to try to formulate the right question, wonderfully difficult to consider alternative hypotheses, wonderfully difficult to use alternative unfamiliar methods to try to figure out which ones are true and which ones are false. And we're in a new field with these particular subset of questions about the adaptive significance of traits that make us vulnerable to disease. I think there's a lot of clear thinking that we all need to help each other to develop in order to make our science progress. And that's the end of what I have to say. Thanks very much. I think I have time for one or two questions, and then it's off to lunch. Please. Very loud.
It seems to me that we're often concerned about wrong hypotheses. You brought up the example of the diabetes as an anti-freezing agent. But in science, there's lots of wrong hypotheses, lots of wrong tests.
Right.
It's been suggested that the majority of findings that are published in biomedical journals are in fact incorrect. And it seems to me that we shouldn't get too off course with that. And in fact, a healthy debate on hypotheses that are supported with some evidence is a good thing.
So Joe's point is there's lots of wrong hypotheses in all science. I'd like to echo that. I mean, you're going to hear people say there are a lot of ridiculous ideas being suggested in evolutionary medicine, and the right answer to that is yes, like there are in all of science, and isn't it wonderful that we can demonstrate why many of them are wrong? And a lot of them turn out to be very hard to figure out whether they're right or wrong, and that's what makes our field fun. Last question.
So we spoke a little bit at breakfast yesterday.
Loud.
We spoke at breakfast, and you asked me to bring this topic up.
Yes, please.
Methodology in evolutionary medicine, and how do we get physicians and medical students to think more about evolutionary medicine? One of the ways might be to couch it in terms of how do we do good science, and to introduce the concept of evolutionary medicine in the context of how do we test hypotheses, how do we know we have the right cases and the right controls. In that way, bringing it.
So isn't Josh's idea lovely? Did you all hear that? He points out that instead of seeing this as some special burden we bear, in fact, if you can teach people to think clearly about how to test evolutionary hypotheses, then they have a real running start at being good critical thinkers about medical hypotheses in general. And he suggested actually that the way evolutionary medicine would fit into the curriculum is in a curriculum about making medical decisions using evidence and proper statistical Bayesian thinking and theory altogether, instead of just the mishmash of thinking that we usually see in physicians nowadays. I'm sorry there's not time for more questions. Next year, it'll be a whole hour for this. Have lunch, and we'll see you back, or we'll see you at your discussions at 1.